Hello there. Welcome to Fundamentals of Biology, Lesson 10, where we're going to be looking at cellular respiration. Now, this follows on from the previous session where we looked at cell membranes and membrane processes. So some of the terms that we looked at in that session, so diffusion, facilitated diffusion, osmosis, and active transport, well, a couple of those terms we're going to see again in today's session. So hopefully you can remember what they are and what they involve. If not, then go back over them again because they are important processes within biology. But like I say, today we're going to look at cellular respiration. We can see that on the diagram on the slide here. It is a fairly complex process, but basically what it does is it takes the food that we eat and the oxygen that we breathe, and it converts it into a usable form of energy inside of our cells. The energy that we're actually going to produce is called ATP, or adenosine triphosphate. Now, you've heard me mention ATP in a few of the other sessions that we've done now. So in this session, we're actually going to talk a little bit about what it is and how the body or how cells produce it. And like I said just now, it comes from the energy that's stored up in our food that we eat, and we use oxygen to unlock the energy that's in our food, and then we can convert it into this usable energy source, which is ATP. So during this session then, we're going to first start out by talking about ATP. So we'll have a look at it and talk about how it actually provides energy or donates energy to specific cellular processes. And then we're going to go on and actually look at the main stages of cellular respiration, specifically aerobic respiration. So hopefully these are terms that you're familiar with. So aerobic and anaerobic. Aerobic means in the presence of oxygen, so with oxygen. So this is what we're going to follow through today. But I will mention as we go through a different version, which is anaerobic respiration. So that is where we don't have oxygen available. So look out for those points. The four main stages of aerobic respiration are glycolysis, and then we move on to the link reaction, and then the Krebs cycle, which is otherwise known as the citric acid cycle. And then we'll finish off by looking at oxidative phosphorylation via the electron transport chain. That is a bit of a mouthful, but hopefully when we get there, we can unpick it a little bit, and then hopefully you'll realize that I certainly find it actually really, really interesting to look at. It's a pretty complex process, but it's fascinating as well. It's one of those things in biology that you look at and you think, how on earth did that ever evolve? It's incredibly complex and but very, very interesting. These are the topics for today. We'll work through them. There's a little quiz at the end just to check how you've gotten on. But with all of the previous topics, this isn't the kind of thing that I would imagine, if you've never done biology before, I wouldn't imagine you to feel really comfortable with it after just a single pass. So obviously we're doing this lecture now, but I wouldn't expect you to get to the end of it and think, okay, that's easy, I've got that now. This is the kind of topic that you do need to go over a couple of times. Maybe a few times, and use quizzes and test yourself and make notes, and it, hopefully by going back over these things a few times, it will help to sink in. So what we can see on the slide here is a molecule of ATP, or adenosine triphosphate. And this is the energy currency of life. So all forms of life, whether you're a bacterium or a human, or a plant or a fungus like a mushroom, you use ATP to carry out cellular work. And it's a high energy molecule that stores energy that we get from our food. And it powers pretty much everything that life does. So it is an incredibly important molecule. So let's have a look at it and break it down a little bit. We've actually seen some of these bits in previous sessions. So if you think back to the session that we did on nucleic acids, so when we looked at DNA and RNA, if you can recall, the monomers of DNA and RNA are called nucleotides. And they were made up of a pentose sugar, a phosphate group, and a nitrogenous base. And we can see the similar things here. So if we look right at the heart of this ATP molecule, we see the pentose sugar ribose. So ribose is the same sugar that we find making up the nucleotides of RNA. So ribonucleic acid. Attached to the ribose, up here, we have a nitrogenous base. We have the base adenine. So again, this is what we find in the makeup of an RNA nucleotide. So ribose and an adenine. Together, these two make up a molecule called adenosine. So together, these make up adenosine. So if we remember the name of ATP, it's adenosine triphosphate. So we have adenosine here. And then if we look over here, we have 3 phosphate groups. So if we just had one phosphate group, that would be a nucleotide. So a nucleotide that could be a part of a strand of RNA. But instead, when we're making ATP, we have that second and then third phosphate group. So how does ATP as a molecule then donate energy to cellular processes? How does it actually drive cellular work? Well, it's all down to these phosphate groups. So if you notice, and again, this goes back to something that we discussed in our one of our early sessions, where I talked to you about electronegativity, and that is the measure of pull that an atom will have on the shared electrons in a chemical bond, like a covalent bond. And if you can remember from that, I mentioned that oxygen is a highly electronegative atom. And that means that when it makes covalent bonds with other atoms, those shared electrons sit much closer to the nucleus of the oxygen, which means that the oxygen side of the new molecule tends to carry a slight negative charge because it's carrying those extra electrons. And so is the case with these phosphate groups. You can see these negatively charged electrons sitting around the edge of the phosphate groups. So all of those phosphate groups are negatively charged. You know yourself that 2 negatives will not attract to each other. They will repel, just the same way that 2 positives will repel each other. A positive and a negative will attract, but 2 positives or 2 negatives will repel. It's similar if you try and hold 2 magnets together with the same poles aligned, you'll feel that push away, that repelling force. So it's the same with these 3 phosphate groups. They do not want to be in this position. They do not want to be this close to each other. The thing that holds them together, though, are strong covalent bonds. So they are essentially forced into this position where they are sitting right next to each other. And what that means is because they have this natural force that wants to push them apart, they want to repel each other, when we look at an ATP molecule, we can see that it has a store of potential energy. All those 3 phosphates want to do is they want to be able to break the covalent bonds that form between them. So we've got these covalent bonds here between them, and they want to break those bonds, and then they would all fly apart from each other. And as they move quickly away from each other, that potential energy will be released. And that's exactly how our ATP molecule actually powers cellular work, by specifically removing this 3rd phosphate group. Covalent bond that holds the 2nd and the 3rd phosphate groups together gets broken apart by an enzyme. So if you remember, that process of using enzymes to break molecules apart is called hydrolysis. So that 3rd phosphate group is broken off. And that's exactly what we can see on this diagram here. On the left-hand side, we've got, so it's arranged in a slightly different way here, but it's the same. Hopefully you can see that. We've got our ribose here, we've got our adenine, and here we have our 3 phosphate groups. And you can see that this, the covalent bond between the 2nd and the 3rd phosphate group is highlighted in yellow there. What will happen is an ATP molecule will attach to, say for instance, a protein, a channel protein within a plasma membrane, like we saw in active transport in the previous session. And that protein will then act as an enzyme to break apart that covalent bond. And as that bond is broken, this third phosphate group will move away. It will repel from these other two phosphate groups. And as it does that, it will release energy. So you can see here it's represented ATP as a fully charged battery because of these high energy bonds between these repelling phosphate groups. You break that bond, the third phosphate group fires off, and that releases energy. And we can see that on the right hand side here. So you can see that this bond is broken, that releases energy, and the cell is able to then use that energy to do work. And that work could be something like muscle contractions. It could be the active transport that we looked at in the previous session. After ATP has given up its energy, so we've broken off that third phosphate group, the energy has been released. What we are left with now is this molecule, adenosine, and then 2-phosphate. So this is adenosine diphosphate. So you can see that up here, and otherwise shortened to ADP. So over here we've got ATP, and then over here we have ADP, adenosine diphosphate, and then we also have this inorganic phosphate, this free phosphate group over here. This kind of creates a bit of an issue for life because we consume and use an awful lot of ATP molecules every single second to power our cellular work. So we need a lot of these, but as soon as they're used, we generate these. So we're just left with ADPs and a lot of these sort of loose independent phosphate groups. How do we get more ATPs? If we had to build new ATP molecules from scratch every time we needed them, we'd pretty much just do nothing but sit around eating the raw ingredients for making ATP molecules, and we wouldn't have any time or energy left to do anything else. So instead, life has evolved a way, an ingenious way of recycling these ingredients. So it will take ADP, and then it will take this third phosphate, and it will attach it back on. So you can imagine, and remember, they don't want to be next to each other. This phosphate group is negatively charged, so are these two. So it's not naturally going to happen. They repel from each other. So you need some kind of energy to actually attach this phosphate onto there to overcome the repelling force. Now, as we've hopefully you can sort of start to see the issue here, because as we've just been talking about, the way that cells do work, the way that they go against the natural order is by spending ATP molecules. But the issue that we have here is you can't spend a molecule of ATP to attach this on here to make a molecule of ATP. If you spend one molecule to make one molecule, you'll never end up with a surplus of ATPs that are available to do any other kind of work. That isn't an option. Instead, life evolved a pretty ingenious but quite complex method for attaching this third phosphate onto this ADP molecule to basically turn it back into an ATP molecule so it can do some more work, so it can do its job again. And that process is cellular respiration. And as we'll look at as we go through, keep that in mind, that the end prize or the end goal here is to stick those third phosphate back onto ADP molecules to recycle them, to recharge them, if you like, into ATP molecules. And what we do is we use our food and then we use oxygen to do that. So that's the main gist of ATP and how it donates energy to cellular processes. What we need to now do is go through this process of recycling the ADP and the phosphate back into ATP, and that is respiration. So cellular respiration then, it's the process by which the chemical energy of our food molecules. So if you remember, we've looked at a lot of these now. So we've looked at the structures of carbohydrates, of lipids, and of proteins, and of nucleic acids. And what we see with those is they're just lots of atoms all joined together, typically through covalent bonds, to make these molecules and compounds. In those bonds, so the bonds between the atoms, there's a lot of stored energy. So if you start to break those bonds apart and then they start to make new bonds with other atoms, you tend to get a release of energy. So what we're going to do through the process of cellular respiration is we're going to take a food molecule in and we're going to start breaking it down and then essentially using the energy specifically that are in the electrons around the atoms. So I want you to keep that in mind as we go through. We'll probably go through some confusing stuff, but I want you to try and keep in mind as we go through that what we are doing is we are breaking apart our food molecules and we are gathering up electrons. So we're harvesting electrons from our food and we're going to put them to work right at the end of the process of respiration. Okay, so if you go to any dark places during this, the happy places, we're just harvesting electrons. So you can keep coming back to that. And then I'll tell you when we're actually going to put those electrons to work. Respiration then is the process by which the chemical energy in food is released and partially captured in the form of ATP. I say partially because as you'll see, as we go through the process, we do generate some waste products as well. So it's not that there are no 100% efficient processes, energy processes out there, and this isn't either. We do generate some waste products, and I'll point those out as we go through. So pretty much all of our food molecules can be turned into ATP through the process of respiration. So we can have a range of carbohydrates, fats, and proteins. But we typically, when we're talking about respiration, we typically follow glucose. And if you remember from our carbohydrate session, I mentioned then that glucose is the preferred source of fuel for life, particularly animals. Glucose is the most commonly used source of fuel. So that's the one that we're going to follow. But just keep in mind that you can also do this with other carbohydrates, but also fats and proteins as well. And what we can see here is the process of respiration, or the majority of it anyway, occurs within the mitochondria. And this is a cell organelle that we looked at in a couple of sessions ago. The mitochondria we find in all eukaryotic cells. So here we can see an animal cell and a plant cell, but also we'll find mitochondria in our protists and our fungal cells as well. And what we can see right at the bottom here is the equation. So this is the balanced equation for cellular respiration. So hopefully you remember that C6H12O6, well, that's the molecular formula for glucose. So 6 carbons, 12 hydrogens, and 6 oxygens. We take a glucose molecule, we take 6 oxygen molecules, and then that converts into 6 CO2 molecules. So CO2 is a waste product of cellular respiration. We get 6 water molecules. So water is also a waste product. And then we get energy. So this is the ATP energy, and also heat, which is produced as a waste product as well, or a byproduct of respiration. The beauty of this equation is that it shows the process of respiration. But if we look at it in reverse, so energy plus water plus CO2 gives oxygen and glucose, well, what we see there is photosynthesis. So photosynthesis uses light energy and water and carbon dioxide to produce sugars, and then oxygen is the waste product. So we find this really nice cyclical motion where photosynthesis produces the ingredients for respiration, and respiration produces the ingredients for photosynthesis. So you can see how these two processes go together. Let's start by having a quick run through of the processes that we're going to look at as we go through the rest of the session then. So we know that what we're doing is we're taking our food and we are using oxygen to essentially convert the energy within that food into ATP. So let's have a quick run through. What you can see here on the diagram, this sort of pink thing here, that is a mitochondrion. So if you remember, mitochondria, they're the powerhouses of the cell, and they are what generates the majority of our ATP. So here's the mitochondria. We can talk more about the structure later on again, but I did cover this a couple of sessions ago. So if you remember, you have an outer membrane, you have an inner membrane. Between the two membranes, you have this intermembrane space. And then inside the inner membrane, we have a substance that is referred to as the matrix. So outer membrane, inner membrane, intermembrane space, and then the matrix. All of those structures are going to become important as we go through this topic. So let's start outside of the mitochondria though. So we're just out here in the cytoplasm of the cell. And we're going to start with our glucose molecule. So we're just going to follow one glucose molecule for now. And we start the process by going through something called glycolysis. Now I'm just going to go through this quite quickly, and then we'll go through it in more detail using the slides to come. So glycolysis then occurs outside of the mitochondria, inside the cytoplasm of the cell. And enzymes will come along, and we also have to invest a little bit of ATP to start this whole process. So we invest 2 ATP molecules, and we get some enzymes acting to break glucose apart. So glycolysis breaks glucose in half. So glucose is a 6-carbon molecule. So remember, it's C6H12O6. We break that in half into 2 pyruvate molecules. So each pyruvate molecule has 3 carbons in it. So that's where our 6 carbons from glucose goes. During the process of breaking glucose apart into 2 pyruvates, we do actually generate a couple of ATPs. We actually generate 4 ATPs, but because we spent 2 to start the whole process, we say that we've actually made 2 ATPs during glycolysis. What we also see are these things called NADH. So NADH is an electron carrier. They actually start life as a little molecule called NAD+. So NAD+ will come along, and during the process of breaking glucose apart, it will gather up a couple of electrons and a proton. It's stripped away from the glucose molecule. Each NADH represents 2 electrons and a proton. These will then carry those electrons and protons into the mitochondria. So if we notice here, we follow all the way along here, and they are going to get fed into the final stage of cellular respiration. So we're going to gather up some electrons. Remember, I said earlier, that is what we're doing. We're breaking apart our food, and we are harvesting up the electrons. So that's glycolysis. We finish it. We've generated a couple of ATPs, and we've also got a couple of pyruvate molecules. We also have created a couple of NADHs, and we also generate a couple of water molecules as well. But there's an important point to consider next, and that is what happens to the pyruvate. Now, if you remember earlier on, I mentioned that what we are going to be following today is aerobic respiration, so in the presence of oxygen. And oxygen plays an incredibly important role in respiration. For pyruvate to enter into the mitochondria, so it's going to move from the cytoplasm of the cell, and it's going to move into the matrix, so into the very center of the mitochondria. For that to happen, we need to have plenty of oxygen available. So oxygen sits in the mitochondria, and it keeps the whole process moving. If you don't have enough oxygen going into the mitochondria, the pyruvate cannot get in there. So instead, what will happen is something called anaerobic respiration. So this is the process by which we can generate a bit of ATP, but without the presence of oxygen. Now, this is good because it means that we can generate some ATP even in low oxygen conditions. And it also means that we can generate some ATP very quickly because this process occurs quite quickly. But there's a real downside to this. So the downside to anaerobic respiration is that this pyruvate, instead of going into the matrix, it stays out here and it gets converted into lactic acid. that will be something that you would have felt if you've ever done any kind of exercise. So even just sort of walking up a set of stairs really quickly or walking up a hill really quickly, you will feel that after a time your legs will start to burn. Or doing any kind of exercise for a prolonged period of time, you will start to get burning and aching muscles. Well, that burning and aching sensation is generated, or it's created, by the buildup of lactic acid. And that is because once we continue to exert ourselves, we reach a limit of how much oxygen we can get to our cells. Once our energy output exceeds our oxygen input, well, then our respiration switches from aerobic, where the pyruvate goes into the mitochondria. It switches from that to anaerobic, where it stays out here in the cytoplasm and starts to generate lactic acid. And that is really painful, and it's not something that you can maintain indefinitely. There will come a time where you need to slow down, or you need to stop whatever it is you're doing, and you need time to build up your oxygen levels again so you can break down and deal with the lactic acid. That's basically what happens when you get that burning and aching muscle feeling from exercise. And interestingly, when we talk about fitness and going to the gym or running or whatever it is to increase our fitness, I think we often think of that as sort of losing fat and gaining muscle. And although that is an element to it, the big part of improving our fitness, particularly when we talk about aerobic fitness, is improving our oxygen delivery system. So strengthening our heart and lungs, sort of improving the speed and the volume of blood that we can get through our system so we can carry more oxygen to our cells. So when we build up fitness, so you typically hear this referred to as doing cardio work. So you're building on improving and strengthening your cardiovascular system, the heart and the lungs and the blood vessels, so we can get more oxygen to our cells more quickly, so we can maintain aerobic respiration and prevent or reduce the amount of anaerobic respiration we're doing, so we can sort of put off the buildup of lactic acid. Because what you'll also see, if you look at the rest of this diagram down here, for each glucose molecule during glycolysis, we only generate 2 ATPs. But as we go through the rest of the processes, you can see over here in oxidative phosphorylation, we're actually generating 26 to 28 ATPs. So we get a lot more ATP production when we have oxygen available. So that's why it's important for us to maintain that level of cardiovascular fitness, if you like, so that oxygen delivery mechanism. Just one last note on this before we move on, is that different organisms will carry out anaerobic respiration in different ways. I've just mentioned that we produce lactic acid. So as animals, we generate lactic acid during anaerobic respiration, so which is sometimes referred to as fermentation. But if you were something like a yeast, or you were certain species or certain types of bacteria, instead of producing lactic acid, instead what you would produce is ethyl alcohol. And this is the basis behind brewing. So when we brew something, essentially what we'll do is we can get sugars from barley and things like that, and cereals, or from grapes. So we can get the sugar that are in grapes to make wine, or from barley to make beer. And essentially what you do then is you take those sugars and you mix them with water and then yeast, and then you put a tightly sealed lid on it to prevent oxygen from getting into it. And then what will happen is the yeast will start converting the sugars into ATP. So they will go through this whole process. So they will go through aerobic respiration until there is no oxygen available to them. And then once all the oxygen's gone from the container that you've put it in, well, then the yeast will switch to anaerobic respiration or fermentation. And that is basically glycolysis without the presence of oxygen. So they are taking sugars, they convert them into pyruvate, and then the pyruvate, because there's no oxygen, they can't get into the mitochondria. So the pyruvate instead gets turned in to ethyl alcohol and carbon dioxide. we also get the carbonation in beer from that, as well as the alcohol. And that's essentially the basics behind brewing. We're taking yeast, we're starving them of oxygen, they're switching to anaerobic respiration, so fermentation, and they're converting the sugars into alcohol and carbon dioxide to make lovely beer. Anyway, that's glycolysis. We're working on the assumption, though, that we do have oxygen present, so our pyruvates aren't going to get converted into lactic acid or ethyl alcohol. Instead, what they're going to do is they're going to move into the mitochondria, into the matrix. And what happens then is we go through the link reaction. So the link reaction just sort of breaks off a carbon and a couple of oxygens from each of our pyruvates. So we actually get a couple of carbon dioxides made here. And then it produces these molecules called acetyl coenzyme A, or acetyl-CoAs. And that then moves into the citric acid cycle, or the Krebs cycle. So notice we've generated a couple of NADHs, those electron carriers. And notice where they're going. They're going right towards the end stage again. Citric acid cycle completes the breakdown of our glucose. So by the time we finish the citric acid cycle, our glucose has gone. All of the carbons have been given off as carbon dioxide. We've also generated some water. The citric acid cycle does create a couple of ATPs, but also we've harvested up quite a few more electrons in our NADH, and also in this second type of electron carrier, which is FADH2. Okay, so FADH2, these are all just electron carriers. They're carrying their electrons to the final stage, which is oxidative phosphorylation via the electron transport chain. And that's basically where we finish our story. So we use the electrons and we use the energy in electrons specifically, and the power of oxygen. So if you remember from a couple of slides ago, I reminded you about the electronegative properties of oxygen. Oxygen exerts an incredibly strong pull on electrons. All of these electrons that are being carried by these electron carriers are going to be really, really keen to fall down towards oxygen. They want to move towards oxygen as oxygen exerts a really powerful pull on them. And that means that those electrons represent potential energy. And we're going to use that energy as the electrons move towards oxygen to essentially turn our ADP and our phosphates into ATP. Okay, so I'm very aware of the fact that I just said an awful lot of words. So I think what we need to do is start to go through each one of these sections, and I'll give you some text on the slides so you can actually see it and you can make some notes. And then we'll go through each step slowly, and then hopefully that will help it to make sense. Okay, let's begin. Oh, before we move on, you can see here from one glucose molecule, we're getting about 30 or 32 ATPs. Not a huge number when you consider that a single muscle cell that's working hard will generate something like 10 million ATPs every single second. So this is a process that's happening incredibly quickly. Let's start by having a look at glycolysis then. So if you remember, this occurs outside of the mitochondria in the cytoplasm of the cell. So in cellular respiration, glucose and other molecules are broken down in a series of steps. And this begins with glycolysis, which happens in the cytoplasm. And what we can see on this diagram is a breakdown of the different processes that are happening during glycolysis. Each one of these processes is being carried out, is being facilitated by specific enzymes. So when you see something that's happening within cellular respiration, if there's any kind of reaction going on, something being made, something being broken apart, you can be pretty sure that there's an enzyme at work there. So let's start off over here with our glucose. So our C6H12O6. We're just following the carbon skeleton because otherwise it can get pretty confusing. So let's follow just the carbon skeleton of glucose. If you remember, I mentioned that we have to spend 2 ATPs, so we have to invest a bit of energy to kickstart this whole process. We start by spending 2 ATPs, and we know what happens to ATP once we've used it. It gets converted into ADP. So with this diagram, the stuff at the top is what goes into glycolysis. The stuff at the bottom here is what comes out of it. So our 2 ATPs are spent, which leaves us with a couple of ADPs. We can forget about those for now. We get a couple of these NAD pluses. So these are going to be the electron carriers. This is the form that they are in before they've picked up their electrons. 2 NAD pluses come along. They pick up their electrons as we start breaking glucose apart. And now they are in their reduced form, as we'd say, they are now NADH. So they're carrying their electrons, and they're going to carry those into the mitochondria. We also then get some ADPs and some phosphates. The 4 phosphates aren't shown here, but we would use those as well. And we would actually generate 4 ATP molecules. Now, this is done through a process called substrate level phosphorylation, but we don't need to worry too much about that for now. So we actually generate or produce 4 ATPs during glycolysis. But if you remember, we spent 2 to kickstart the whole process. So we actually only generate, or we get a gain of 2 ATPs. We made 4, but we had to spend 2. And then we also get these 2 pyruvate molecules, these 2 3-carbon molecules. So pyruvate is properly called pyruvic acid, but we typically just shorten it to pyruvate. So that's basically glycolysis. We start off with glucose, we spend a couple of ATPs, we've got a lot of enzymes at work here. The ATPs and the enzymes go to work to break glucose in half. NAD pluses come along, harvest up some electrons. During the process, we're also able to generate some ATPs, so we get a gain of 2. And what we're left with then are 2 pyruvate molecules. So we can look at another diagram to look at this in a slightly different way. We would say that glycolysis can be broken into an energy investment phase because we are spending 2 ATPs to get it started. So we're investing some energy, and that's what we can see here. Glucose comes in, 2 ATPs are used up to start the process. Here you can see the 2 ADPs and 2 phosphates that are left over. That enables us to move into the energy payoff phase, where we actually generate 4 ATPs. And here we can see our 2 NAD pluses coming in and then generating those 2 NADHs. And then we also then finish with 2 pyruvates. And we also generate a couple of water molecules as a waste product. So we can summarize glycolysis with this bottom part of the diagram. Glucose, so the carbon skeletons and the hydrogens and the oxygens, are basically converted into pyruvates, and a couple of water molecules are given off as waste. We are actually generating a gain of 2 ATPs, so we made 4, but we spent 2, so we get a gain of 2 ATPs, and we've also harvested up some electrons in our NADHs. So this is what we finish glycolysis with: 2 pyruvates, 2 ATPs, and 2 NADHs. And we've also generated some water as a waste product. Okay, so that's glycolysis. Hopefully that's not too bad. And if you remember from a couple of slides ago, in the presence of oxygen, so whilst hopefully you're all sat there and you're calm now, so you are getting plenty of oxygen to your cells, well, what's happening now are those pyruvates will be moving in to the mitochondria. If, however, you're watching this whilst you're on an exercise bike and you're struggling for breath, well, the chances are you'll probably be doing fermentation, so the pyruvate will be staying in the cytoplasm, getting converted into lactic acid because of a lack of oxygen. But anyway, good work for riding the exercise bike. So anyway, we're going to work on the assumption that we have oxygen present. So we're going to now follow these pyruvates into the mitochondria. And we can see that on this part of the diagram here. So we're dealing with two pyruvates from our glucose molecule. We're just going to follow one here because it keeps it nice and easy. We know that our pyruvates contain those 3 carbons. Glucose was a 6-carbon molecule. We've broken it in half. So we've got 2 3-carbon molecules, which we can see here. So this is pyruvate. This is the inner and outer membrane of the mitochondria. So you can see here we've got a transport protein. So we are moving pyruvate into the matrix of the mitochondria. So in the presence of oxygen, pyruvate enters the mitochondria, and it's often referred to as the link reaction because it links our pyruvate, or it links glycolysis to the citric acid or the Krebs cycle. So that's why we call it the link reaction. So let's just have a little look at what happens. Pyruvate enters into the matrix, and then enzymes set to work on it, breaking off this part of the molecule that you can see highlighted in grey here. So a carbon with 2 oxygens, and that makes a carbon dioxide molecule. So remember, one of the waste products of respiration is carbon dioxide. This is where the carbon dioxide that we exhale comes from. So it comes from the link reaction, and then also the next stage, which is the citric acid cycle. So we can see 1 carbon dioxide given off here. NAD plus will then come along during this process, and it will harvest up a couple more electrons. So here we can see NAD plus turning into NADH, and then we've got this spare hydrogen ion, and we're going to see these later on as well. And they go off to the final stage, oxidative phosphorylation. And then what this now creates is a 2-carbon molecule. So we've broken off a carbon dioxide from our pyruvate. This leaves us with this 2-carbon molecule. And a 2-carbon molecule is typically referred to as an acetyl group. So we're left with our acetyl group, and then we get this coenzyme A that comes along. So coenzymes are essentially little molecules that will carry substances or other molecules from one part of a chemical reaction to another. So I always tend to think of them as a bit like a taxi service. They'll pick something up as it leaves one chemical reaction, and it will drive it to the next part of the chemical reaction that it needs to go through. So what coenzyme A does is it will attach to our acetyl group that's left from our pyruvate, and that becomes this molecule. So acetyl coenzyme A, or acetyl-CoA for short. Don't go worrying too much about this. The coenzyme A and the acetyl-CoA is basically there just to link glycolysis to the citric acid cycle, which comes next. The key thing to know about the link reaction really is that we lose some of the structure of our glucose, so carbon dioxide is given off, and we've harvested up some more electrons. Once we finish the link reaction, so this is what's left of our pyruvate. So remember, we've got 2 of these. So we had 2 pyruvates left over from our glucose. Once they are in this position, we can then start on the next stage, which is the citric acid cycle. Okay, so this brings us on to the citric acid or Krebs cycle. And this is one of those frustrating things in biology where we've got 2 names for exactly the same process. It's called the citric acid cycle because, as we'll look at on the next slide, citric acid is the first product that's produced during the cycle, but it's called the Krebs cycle because of the chap who discovered it, a scientist called Hans Krebs. These names are used interchangeably and about equally as often, so it's a bit frustrating, but either name is absolutely fine to use. So my suggestion would be to maybe pick one and stick with it. I'd say the Krebs cycle is probably used more commonly. Either one is fine. So let's have a look through. It's a pretty complicated process, but fortunately we don't need to worry too much about the intricacies of each stage of the cycle. Instead, what we want to focus on is what's being generated during the cycle. The Krebs cycle occurs in the matrix, so it's inside the mitochondria, and it generates a lot of chemical energy. So what we're actually doing now is we are completing the breakdown of our glucose, essentially. So remember, glucose started outside of the mitochondria. It went through glycolysis, so it broke in half, turned into two pyruvates. The pyruvates then entered into the mitochondria. We went through the link reaction, which broke off a couple of carbons to make two carbon dioxides off of our pyruvates. They then went through, from the link reaction, turned into acetyl-CoA, and now they get fed into the citric acid or Krebs cycle. And what the Krebs cycle will do is it will complete the breakdown of the carbon skeletons, if you like, of the actual structure of what was originally our glucoses. And what we're left with at the end of the Krebs cycle, you can see in brackets up here, we actually generate a couple of ATPs, but also importantly, we're harvesting up more electrons. So as we break apart what's left from our pyruvates, we're going to harvest up some electrons in the form of NADH and FADH2. So remember, these are our electron carriers. And then they will carry those electrons to the final stage, which is oxidative phosphorylation. So I'm going to show you the Krebs cycle on the next slide. And it's an 8-step cycle, and each step is catalyzed by a specific enzyme. But like I said, well, I'll show it to you because I think it's useful to see it, but you don't need to get too worried about each of the steps and each of the enzymes that carry out those steps. The key thing is to focus on what's actually generated, and it's quite useful to see where. So the NADH and FADH2 produced by the cycle will carry their electrons to something called the electron transport chain that we'll look at next. So let's just have a look at this diagram here first before we move on to the next slide. And this shows what we've just looked at basically. So here you can see one pyruvate. We've been through glycolysis. We've turned our glucoses into two pyruvates. We're just following one at the moment. So one pyruvate will come into the matrix. This is the link reaction here that we've just looked at. So NAD plus comes along, gathers up a couple of electrons, so it turns into NADH. We break off a carbon dioxide. So there's the carbon dioxide. That leaves us now with a 2-carbon molecule, so that acetyl group, which is attached to this coenzyme A to make acetyl-CoA, and then that feeds into the Krebs cycle. And you can see that represented by the cycle there. And remember, the top part of the diagram is what feeds in, and at the bottom, this is what we're interested in. This is what's generated through the Krebs cycle. So for each pyruvate that feeds into it, we generate 3 NADHs, we generate 1 ATP, and we also generate 1 FADH2. And as a waste product, we actually produce those 2 molecules of carbon dioxide. If we follow the carbons through them, we can see where they go, where they disappear to. We follow one pyruvate, so each grey ball there is a carbon. They come in. One carbon is given off in the link reaction as carbon dioxide, and here are the remaining 2 carbons. They go into the Krebs cycle, and here are those 2 carbons being given off as CO2. So by the end of the Krebs cycle then, all of our glucose molecule that we started off with has now disappeared. It's all been broken apart. We've given off the carbons as carbon dioxide. We've generated a little bit of water. But importantly, we've harvested up a lot of electrons through our NADHs and our FADH2s. What we can see here then is what's generated during the Krebs cycle. And remember, this is just following one pyruvate. So for one pyruvate, we get 3 NADHs, 1 ATP, and 1 FADH2. But remember, we get 2 pyruvates from our original glucose molecule. So the Krebs cycle in total will produce 6 NADHs, 2 ATPs, and 2 FADH2s, and then 4 carbon dioxide molecules as waste. And then that will basically enable us to move on to the final stage. But first, let's have a quick look at the Krebs cycle. So this is what it actually looks like. So like I said, don't get too bogged down by each of the stages. What we want to look at is where we're getting our products from. So this is our acetyl-CoA. This is what was produced during the link reaction. So here we can see the 2 carbons. that will get fed in to the cycle. So it gets fed in by joining the acetyl group to this little molecule called oxaloacetate. That will then turn it into this product, which is called citrate or citric acid, hence the name citric acid cycle. And then what happens then is we go through all of these different stages, all catalyzed by different enzymes, and it's essentially manipulating and breaking apart the molecule to generate certain products. So what we can see here is we break off a carbon dioxide from that molecule and we generate an NADH, so some electrons are harvested. We then move around to the next stage and we break off the second of our carbon dioxides, and we also harvest up some more electrons during that stage. We then move around here and we're generating a little bit of ATP, so 1 ATP per acetyl group, so 2 in total. And then we move around to the next stage and we harvest up a couple more electrons in the form of FADH2. And then we move around to this final stage where we harvest off some more electrons in the form of NADH. If we come back here, you can see though for each pyruvate, which gets converted into acetyl group, for each one of those we get 3 NADHs, 1 ATP and 1 FADH2. And we generate 2 carbon dioxides. So let's go forward. There's 1 NADH, there's 2 NADHs, there's 3 NADHs, there's our 1 FADH2, there's our 1 ATP molecule, and there's 2 carbon dioxides. So there we can account for all of the products that we get from the Krebs cycle, as well as the waste products in the form of CO2. So that's essentially the Krebs cycle. It completes the breakdown of what was originally our glucose molecule. We have now given off CO2. So remember, this is where the CO2 that we actually exhale, this is where it comes from this process. That's where the carbon skeletons have come from our food. We've given that off. We've generated a small amount of ATP, so 2 in glycolysis and now 2 during the Krebs cycle. And we've harvested up a lot of electrons during all of those stages. The next thing we have to do is we're going to put those electrons to work. We're going to do that during the process of oxidative phosphorylation via the electron transport chain. So I know it's a bit of a mouthful, but we'll break it down step by step. For me, this is one of the most fascinating topics that we cover in all of our sessions, because it's just fascinating. It's really sort of convoluted and a bit tricky, but it's an amazing feat of evolution, really, in my opinion. What you can see down here in this diagram is the basic diagram of the mitochondrion. You can see the outer membrane, you can see the inner membrane, and you can see here that the electron transport chain, which I'll show you on the next slide, is actually embedded within the inner membrane. So the inner membrane of the mitochondria plays a pivotal role in oxidative phosphorylation, as does the gap between the membranes, what we call the intermembrane space. Okay, so I'll use that term a bit as we go through this. So oxidative phosphorylation uses the oxygen we breathe. So I haven't really talked much about the oxygen that we breathe yet, but this is where it becomes important. So this process uses the oxygen we breathe and the electrons that we've harvested during glycolysis, the link reaction, and the Krebs cycle, and it's going to use that to generate our ATP, to convert ADP and phosphate into our ATP molecules. Following glycolysis in the Krebs cycle, NADH and FADH2 account for most of the energy extracted from food. So I say most because we've also given off some energy as heat during the processes, but also we've given off some water and also some carbon dioxide. But the electron carriers now are going to transport and donate their electrons to the electron transport chain that is embedded within that inner membrane of the mitochondria. So when we look at the inner membrane of the mitochondria, what we can see is a bit like the plasma membrane that we looked at in the previous session. It's made up of phospholipids, and those phospholipids are embedded with a whole suite of different proteins. The electron transport chain is just a network of proteins that you can see here. So they can make up some quite big complexes of proteins. So let's follow it through step by step. So electrons are transferred from NADH and FADH2 to the electron transport chain. So what you can see up here is NADH is coming in and it's dropping its electrons up here. Here's FADH2, it drops its electrons down here. And then the electrons are passed through a number of proteins, including these little ones called cytochromes that basically shuttle the electrons between the different proteins. And ultimately, they pass them down to oxygen. So let's talk about that, because this is what it's all about, really. So the electrons are dropped up here. They're passed into those into those proteins. The oxygen that we breathe, you can see it down here, this little red circle down here. And oxygen sits at the bottom of the electron transport chain. So it just sits down at the bottom of this last protein. Now, if you remember what I mentioned at the beginning of this session, oxygen is highly electronegative. It will exert a huge pull on electrons. So when electrons are far away from oxygen, they represent potential energy because what they want to do is essentially fall towards oxygen. So I always think of this as a ball rolling down a hill. So if you put a ball at the top of a hill, so if we think of our electron transport chain as a hill and our electrons are a ball, well, what's going to happen if you drop that ball at the top of the hill? It's just going to roll down, isn't it? The ball will roll down the hill. And it's going to do that because of gravity. And we're familiar with gravity. We deal with it on a day-to-day basis. You know, you want to pick up a glass to drink from it, you have to fight against gravity. We have to exert energy to do that. We're constantly aware of the effects of gravity pulling things downwards. And when something is, so a glass is on a table, for instance, it has potential energy. That glass could fall off the table and smash on the floor, and then it would have less potential energy because it's closer to the ground. And it's a similar way with electrons and oxygen. Electrons would represent our ball, and oxygen would represent gravity. So oxygen is pulling those electrons down towards it. And when the electrons are up here and oxygen is down here, the electrons have potential energy, just like the glass on the table or the ball at the top of the hill. And what they want to do is they just want to go straight down to oxygen. And as they do that, as they get closer to oxygen, they give off that potential energy. That energy that they've stored or they have stored up here is given off. Now, if NADH and FADH2 just came into the matrix, so this is in the matrix here, this is the intermembrane space, so these proteins are embedded within the inner membrane of the mitochondria. But if the electron carriers just came in here and dropped their electrons off, those electrons would have nothing to stop them from going straight down to oxygen. And as they did that, they would give off that energy, they would release that energy in one huge amount. And essentially what would happen is we would all go poof in a big puff of flames, as all of that energy was released instantaneously. And clearly that's not going to be conducive to longevity of the species or particularly the individual. I would say that this is akin to going to the petrol station in your car, filling the tank full of petrol, and then lighting a match and sticking the match in the petrol tank. All of the stored energy in that petrol would be released instantaneously as your car goes boom into a puff of flames. That obviously isn't very useful. Instead, what a car does is it gently and slowly releases the energy in your fuel bit by bit to power the engine which turns your wheels so you can actually do useful work with it. And this is similar situation to what we have here with the electron transport chain. These electrons, instead of just falling straight down to oxygen and releasing all of their energy instantaneously, instead they get passed into these proteins. And then they get passed from protein to protein. So they get this very slow fall down towards oxygen. And each time they take a step closer to oxygen, they actually give off a little bit of their energy that they have when they're up at the top here. So as they get closer to oxygen, they have less and less potential energy because they are giving that energy off as they move down. So what we actually say is that the electron transport chain breaks the large free energy drop from the electrons from our food to oxygen in small steps that release energy in manageable amounts. Now, importantly, that this isn't, when I'm talking about energy here, I'm not talking about ATP. So we don't generate any ATP during the electron transport chain. We're on a much smaller scale than that. So we're just dealing with electrons. But what they actually do, so as these electrons move down towards oxygen, the energy that they give off is it basically turns these proteins that make up the chain into pumps. And what we'll see on the next slide is that in the matrix, so this is all the matrix here in the middle of the mitochondria, all of those hydrogen ions that we saw generated during glycolysis, the link reaction in the Krebs cycle, well, all of those hydrogen ions are inside the matrix here. So remember, a hydrogen ion is just a proton. So a hydrogen atom, so you remember from our first session together, is just a proton with an electron moving around it. Well, you separate the electron and the proton, what you're left with is that single proton, which we refer to as a hydrogen ion or a H+. So there are loads of these hydrogen ions inside the matrix. As the electrons give off their energy as they move down towards oxygen, that energy is used to pump the hydrogen ions from the matrix up here into the intermembrane space, so the space between the membranes of the mitochondria. We'll get onto that on the next slide. What you can see here then is when the electrons finally emerge from the bottom of the electron transport chain, they combine with a couple of the hydrogen ions down here. So one proton and one electron gives you an atom of hydrogen. They will then combine with the oxygen that sits down here, and that generates water, so H2O. Two hydrogen atoms and an oxygen atom gives you water. So this is also, remember, we generated a bit of water during glycolysis, but we also get some water generated from the electron transport chain here. So this is what the whole process essentially looks like. In this, we can see it in this diagram. So let's follow it through. So here we've got the inner membrane of the mitochondria. So you can see it's a phospholipid bilayer with the proteins that make up the electron transport chain embedded within it. This lighter color on the inside is the matrix. So this is the middle of the mitochondria. This darker orange color here is the intermembrane space, the space between the inner and the outer membranes. So here's NADH and FADH2 dropping off their electrons. Here's oxygen sat at the bottom of the electron transport chain. As the electrons move down the electron transport chain towards oxygen, they give off their energy, and that energy is used to pump hydrogen ions out here into the intermembrane space. What that ultimately leaves us with, so once our electrons have moved out of the electron transport chain, they've combined with oxygen and some hydrogen ions, generating water as a waste product. So that's gone, that's done. What we're now left with is an abundance of hydrogen ions out here in the intermembrane space. So we've got a big buildup of hydrogen ions out here. Now, remembering what we covered in last week's session, we can use some of the terminology that we talked about then. So we've got a lot of hydrogen ions out here, not very many left in the matrix because we've just pumped them all out here. So what we have created there is a concentration gradient. So those hydrogen ions are going to want to diffuse down their concentration gradient until they are evenly distributed on both sides of the membrane. But there's a problem. So the hydrogen ions cannot, because they are positively charged, they cannot get through the phospholipid bilayer. They cannot get back through the proteins that they were just pumped through. So instead, what they have to do is go through the only channel that is available to them, and that is this special protein complex here called ATP synthase. So ATP synthase is the only avenue for these hydrogen ions to diffuse through. So it's a form of facilitated diffusion that we talked about in the previous session. So what's going to happen is the hydrogen ions will start to diffuse back through ATP synthase. And I'll show you a clearer diagram of this on the next slide. But ATP synthase essentially works like a rotor. This is very similar to the way a hydroelectric dam works. So with a hydroelectric dam, you store lots of water behind a wall. The water can't get through the wall until you open certain sluice gates and things like that. So you open the gates and then water can flood through, and then it goes through a turbine, turning the turbine, which generates electricity. Well, this works in a very similar way. Instead of water and a wall, we've got the phospholipid bilayer, we've got hydrogen ions, and instead of a turbine, we have ATP synthase. So hydrogen ions will diffuse through ATP synthase, and the flow of these hydrogen ions, just like the flow of water, will generate enough force. It's something that we call proton motive force. So we're dealing with protons here, those hydrogen ions. The flow of those protons will essentially turn a rotor that is inside this protein, and that generates enough force to take in ADP and our phosphate and join them together to make a molecule of ATP. So that is essentially the whole point of what we've done up to this stage. So the whole point of glycolysis, the link reaction in the Krebs cycle, was to generate a little bit of ATP, but really it was to harvest up those electrons. Those electrons can then be used to create a concentration gradient of hydrogen ions, which will then flow through ATP synthase, generating enough force to overcome the repelling force of the phosphates in ADP and this third phosphate group. Remember, they're all negatively charged. They don't want to be next to each other. So if you want to plug that third phosphate onto ADP, you need a certain amount of energy to do that. And we can't spend ATP to make ATP. So instead, we use the force generated through the flow of hydrogen ions to take that phosphate and plug it back onto ADP to generate ATP. So that is the process of phosphorylation, this putting the phosphate back onto ADP to make ATP. And we are using the flow of hydrogen ions to do that. Where did we get the concentration gradient of hydrogen ions from? We got them from pumping hydrogen ions against their concentration gradient, thanks to the release of energy from electrons as they move down towards oxygen. Where did we get the oxygen from? We breathe it in. Where did we get the electrons from? We got them from our food. So we broke down our food molecules to get those electrons. So hopefully, I know this is it's a little bit of a complicated process, so it's going to be worth going over a couple of times. I've got an animation to show you in a minute, which might help. But here's that close-up sort of diagram of what ATP synthase looks like. So it's just a protein complex. So up here you can see the intermembrane space. Here's the inner membrane, that phospholipid bilayer, and here's the matrix. These little dots, these little circles, are the hydrogen ions, and as they start diffusing back across the membrane, they flow through ATP synthase, and that causes the flow of the hydrogen ions causes ATP synthase to start to spin, to start to rotate. And what that does is you generate enough force to take in ADP and phosphate, attach that phosphate onto ADP to make ATP. So as I mentioned just now, this is something that is often referred to as proton motive force. We're using the force generated by the flow of protons to phosphorylate, to put that third phosphate onto ADP. So let's just put some text to that if you wanted to write down some notes, because I've realized that I've just used diagrams there quite a bit. Electron transfer in the electron transport chain causes proteins to pump hydrogen ions from the matrix to the intermembrane space. Hydrogen ions then move back across the membrane, passing through channels in ATP synthase. So this is a form of facilitated diffusion. ATP synthase uses the flow of hydrogen ions to drive the phosphorylation of ATP. So remember, that phosphorylation, that just means taking that third phosphate and sticking it onto ADP to make ATP. So you might see this referred to as chemiosmosis. So if you remember from the previous session, osmosis is the diffusion of water across a selectively permeable membrane. Well, here we have the diffusion of hydrogen ions across a membrane. So that's sometimes referred to as chemiosmosis. So the use of energy in a hydrogen ion gradient to drive work. So let's do a quick summary. During cellular respiration, most energy flows in this sequence. So we started off with glucose, with all of the energy locked up in the bonds between the carbons, hydrogens, and oxygens. We then relied on enzymes to start breaking glucose apart, and then we were able to gather up or harvest up some electrons that were picked up by our electron carriers, so NADH, and also in FADH2. They carried and donated those electrons to the electron transport chain, which is in the inner membrane of the mitochondria. That energy then was used to create a hydrogen ion or proton gradient. Those hydrogen ions or protons then diffused back across the membrane through ATP synthase, generating this proton motive force that was used to generate ATP. So that's the basic flow of energy through the process of respiration. But interestingly, only about 40% of the energy that was in that glucose molecule is converted into ATP during respiration, and it makes about 32 ATPs. So what happened to the rest of it? What happened to the other 60% of the energy in our glucose molecule? We know that we gave off some carbon dioxide and also some water, so some energy would have been given off there. But also we generate quite a lot of heat during this process. So cellular respiration is quite an inefficient process in the sense that it generates a lot of waste heat. And some organisms, some animals particularly, have evolved a way to benefit from this heat. And we are amongst those animals. So when we talk about warm-blooded animals, the correct term for a warm-blooded animal is an endotherm. So endo referring to inside and therm referring to heat. We generate heat from inside to maintain a nice constant body temperature of between 36 and 38 degrees Celsius. We've talked about in previous sessions where we looked at proteins and enzymes, why it's important for us to maintain that temperature. So how do we do it? We do it through generating heat through our metabolism. So when we're talking about metabolism, we're talking about the rate at which we convert food and oxygen into energy and heat. So let's watch this animation then. She does move quite quickly, so this might be something that you want to go back over and watch a couple of times. But I think it can be quite useful to actually see the animation of how it all goes together.
As this mountain biker heads up the trail, the breakfast he ate this morning is being burned to power his bike ride. His breathing rate increases as his leg muscles demand more oxygen to burn more fuel. Let's zoom down to where this fuel is burned, our cells. Here, the blood vessel on the left delivers fuel and oxygen to a single muscle cell. In cellular respiration, energy and fuel is converted to ATP, shown here as starbursts. Most ATP is made in the cell's mitochondria. ATP powers the work of the cell, such as contraction. Let's take a closer look at how ATP is produced from a molecule of glucose, our fuel. Only the carbon skeleton is shown to keep things simple. The first step is called glycolysis, and it takes place outside the mitochondria. To begin the process, some energy has to be invested. Next, the molecule is split in half. Now, the molecule NAD+, an electron carrier, picks up electrons and hydrogen atoms from the carbon molecule, becoming NADH. Keep track of the electron carriers. They play an important role by transporting electrons to reactions in the mitochondria. In the final steps of glycolysis, some ATP is produced, but not much. For every glucose molecule, only 2 net ATPs are produced outside the mitochondrion. However, glycolysis has produced pyruvic acid, which still has a lot of energy available. Let's follow this pyruvic acid molecule into a mitochondrion to see where most of the energy is extracted. As the molecule enters the mitochondrion, 1 carbon is removed, forming carbon dioxide as a byproduct. Electrons are stripped, forming NADH. Coenzyme A attaches to the 2-carbon fragment, forming acetyl-CoA. Coenzyme A is removed, and the remaining 2-carbon skeleton is attached to an existing 4-carbon molecule that serves as the starting point for the citric acid cycle. The new 6-carbon chain is partially broken down, releasing carbon dioxide. Several electrons are captured by electron carriers, and more carbon dioxide is released. The carbon dioxide that you exhale comes from the reactions of cellular respiration. 2 ATPs are produced by the citric acid cycle for each molecule of glucose. At this point, only a small number of ATPs have been produced. However, more energy is available in the electrons that are being transported by electron carriers. While the citric acid cycle starts another round, let's follow an electron carrier to the next step in the process. Electron carriers such as NADH deliver their electrons to an electron transport chain embedded in the inner membrane of the mitochondrion. The chain consists of a series of electron carriers, most of which are proteins that exist in large complexes. Electrons are transferred from one electron carrier to the next in the electron transport chain. Let's take a closer look at the path electrons take through the chain. As electrons move along each step of the chain, they give up a bit of energy. The oxygen you breathe pulls electrons from the transport chain, and water is formed as a byproduct. The energy released by electrons is used to pump hydrogen ions, the blue balls, across the inner membrane of the mitochondrion, creating an area of high hydrogen ion concentration. Hydrogen ions flow back across the membrane through a turbine. Much like water through a dam, the flow of hydrogen ions spins the turbine, which activates the production of ATP. These spinning turbines in your cells produce most of the ATP that is generated from the food you eat. The process you've just observed, cellular respiration, generates 10 million ATPs per second in just one cell. That ATP can power a biker up the trail, or it can power your brain cells as you learn challenging biology topics.
So let's, as always, finish with a quick recap. So I've got 5 questions here. As always, I'll read through the questions first. Then we can pause for a bit, give you a few minutes to answer the questions, and then we'll go through the answers together. So question number 1, what are the 4 stages of cellular respiration? Number 2, which stage generates the most ATP? Number 3, what is needed for pyruvate to enter the matrix? Number 4, what are NADH and FADH2? And then number 5, what are the 2 main waste products of cellular respiration? There's also a third one there that you can mention as well if you like. So those are the 5 questions. I'll give you a few minutes to answer them, and then we'll go through the answers together. Okay, welcome back. Hopefully you managed to answer all of those. So let's go through the answers. So number 1, what are the 4 stages of cellular respiration? Well, they are in order: glycolysis, the link reaction, the citric acid cycle or Krebs cycle, and oxidative phosphorylation. If you put oxidative phosphorylation via the electron transport chain, bravo. Number 2, which stage generates the most ATP? Well, it's oxidative phosphorylation. Number 3, what is needed for pyruvate to enter the matrix? It's oxygen. Remember, if oxygen isn't present, then pyruvate gets converted into lactic acid, or in yeast, it would get converted into alcohol. Number 4, what are NADH and FADH2? They are electron carriers. So they harvest up those electrons and carry them to the electron transport chain. And then number 5, what are the 2 main waste products of cellular respiration? I did mention that there's a third one as well, really, but that's more of a byproduct. But that's water and carbon dioxide. So we consider water and carbon dioxide to be the true waste products of respiration, and heat is just a byproduct of the process. So hopefully you did okay with those questions. Like I've said several times now, I do not expect you to have a good grasp of this after a single pass through it. This is one of the more complicated topics that we do all year. It is certainly worth taking the time to go back over it. I think it's a good rule of thumb that for each lesson that we do, you should be looking to do an additional 3 hours of reading. So that could be 3 hours of going back over this video, reading a chapter in a good textbook like Biology: A Global Approach, sort of making notes, doing a quiz, answering questions. You can find plenty of quizzes online. So it's good practice to get into. So for each lesson, try and aim to spend an additional 3 hours of additional reading. That will really help to make things sink in. So that brings us to the end of this session then, where we've talked a little bit about ATP at the beginning, and then we focused on cellular respiration, so glycolysis, the link reaction, the Krebs cycle, and then oxidative phosphorylation via the electron transport chain. So hopefully that was useful, and I will see you in the next session. Thanks very much.